So to give you an idea that should really truly be interdisciplinary, we have here Natalie Phillips from the English department, who's going to show you some work from her group, and I think some associated groups, on what happens in the brain when you read Jane Austen.
So thank you so much for having me. Yes, so as mentioned today, I will be, sorry? Yes. Better. Yes, can you hear me?
Yeah.
Excellent. So today I'll be talking about an interdisciplinary experiment that I've been working on at MSU and at Stanford. A study of literary reading that uses technologies from neuroscience to explore the cognitive dynamics involved when we read a work of literature with two different levels of attention. And here, what they're reading is a novel by Jane Austen. And the study is unique in a number of different ways. But as you might imagine, what you just said, one of the things that makes it unique is that I'm the PI, and I'm an English professor, with a co-PI in neuroscience, and collaborators that range from linguistics to MR physics. And part of what I'd like to suggest for today, as part of this broad vision, is that these new kinds of collaborative experiments actually open up critical new ground for mapping complex brain patterns involved in advanced cognition, artistic engagement, and the neuroscience of reading in particular, as well as pushing us to integrate technologies and methods from the humanities with cognitive science, computer science, and potentially down the road research of AI. So our study in particular uses a combination of fMRI and fMRI-compatible eye tracking to investigate neural differences between two particular types of attention, known as close reading and pleasure reading. And what they're actually reading in the study is actually Jane Austen's Mansfield Park. So since it's quite possible, I think, for this audience that many of you were not English majors, I wanted to take a moment to sort of define the term. So close reading, as we talk about it in the profession, asks you to look at a novel's structure, analyzing it and deconstructing it as you go. So you sort of imagine this mode of analysis as looking at a film with a director's eye, for example. We compare this to pleasure reading, which allows you, emotive reading, which allows you to become immersed in a novel or sort of lost in a good book, in a far more relaxed mode of attention. Comparing these actually lets us investigate a key skill taught in English classrooms across the country, the ability to read critically, paying attention to the various structures like plot, characterization, etc., that in fact give a novel its power and depth. It also, for the neuroscientists in the room, lets us investigate what's known as a mode-specific active voluntary and directed attention, in other words, top-down processing of attention-directed cognition. So one of the primary goals of the study was quite simply to investigate the cognitive dynamics and value of studying literature, aside from continuing to produce good writers and thinkers, which I hope we already knew. We wanted to see in any way, shape, or form how this training engages the brain. One of our second goals, of course, was to explore the cognitive dynamics and influences of the different kinds of focus we can bring to reading, particularly novel reading. And in this, we're bringing together neuroscientific research on attention and reading, which are often, in fact, kept separate, to produce a richer picture of natural reading processes and thus of advanced cognition in the brain. So I'm just going to give you a quick experimental walkthrough to just give you a sense of what happens. So basically, a subject who is well trained in this technique, close reading, a literary PhD candidate from Stanford, Berkeley, or San Jose State, comes into the lab and they begin reading the first chapter of Mansfield Park. And we had 12, just to say, we had 12 for our pilot and 18 for the final experiment. And also, we chose PhD candidates because they're fantastic close readers. And as, in fact, we learned from our pilot, English professors were out. We've all forgotten, it seems, how to read for pleasure. So basically, when the subject first comes in, right, they read the entire first chapter outside the scanner. And this is actually to allow them to get used to Jane Austen's style, but also so we can get a sense of how fast they read when they're not inside a loud magnet. Then the student, in fact, goes into the MRI scanner, how happy we all are, And they move it actually through the entire second chapter of Man's Part, paragraph by paragraph, moving at their own speed. This is actually critically important because it's allowing us to do a subject-based design that gets closer to natural reading processes. And throughout, we use a number of experimental technologies that I mentioned, I think in this room, right? I don't need to go through and explain, but we're using fMRI, right, functional magnetic resonance imaging, which gives us a map of blood flow, in particular to the brain, right, which is correlated with neural function. Also, though, and most importantly and uniquely, right, using fMRI-compatible eye tracking, which is allowing us to get a sense of attention-oriented eye movements over time and correlate these back as attentional measures with the brain images that we're acquiring. And alongside these, we're doing very basic measures, heart rate, respiration, etc. So, just to give you a sense, each block, about 9 paragraphs long, they're instructed to either read normally or to close read, right, analyzing literary structures, characterization, syntax, thought, aspects of literary form, and as I said, they can move as long as they want. They can take as long as they want, and moving from slide to slide, reading at their own pace, and moving through paragraphs. Sort of as follows. I just wanted to give you a sort of image of the basic experimental design in scientific mode. Obviously, there's a pause to clear the cognitive noise, but I wanted to just take you through it, and you won't have time to actually read at your own pace, just to give you a sense of how the experiment actually works. This is an example of pleasure reading. And now we switched over, it goes on for longer, but we switched over to the close reading. And of course, at the end, they get out and we ask them to actually write a short literary essay in response to the sections that we've asked them to read for literary analysis, as well as asking them to do a standard set of surveys, what's known as a transport scale. And the final bit, just to give you the sort of scientific basis, right? And of course, right, we have 2 groups, and we flipped the order of the close reading and the pleasure reading to make sure that it's not the text that's going to cause this kind of response, rather than the attention. Right. So I'm just going to actually be talking about one of the things about this work is it's really in the earliest stages. So I wanted to sort of pause and give you some context to explain why we're finding thus far at this early stage is so surprising. So when I began this work, I talked with a number of colleagues, Anthony Wagner at Stanford, Rebecca Sachs at MIT, and joined the Organization for Human Brain Mapping. They were all surprised to see an English professor too. But the expectation from everyone involved was that we were going to see really, really subtle differences in brain activity. And that was because everyone was doing the same thing, i.e. they were all reading Jane Austen, just in 2 different ways. And so because of this, we almost actually did tests where we just zoomed in on very, very specific brain regions associated with selective attention and attentional control, particularly the dorsolateral prefrontal cortex. Just so we'd have a chance of seeing this. And as it turns out, thank goodness we didn't, because the study had never been done before, we just decided to do whole brain imaging. And so I just want to talk about the biggest sort of surprises thus far. So one might have expected to see, for example, pleasure centers activating for the more relaxed style of reading, and a number of other things, but in regions associated with work, attention, and cognitive load at work for literary analysis. But what we're actually seeing is something else entirely. And what caught our attention, and those of my colleagues at Stanford, was how much diverse regions across the brain seem to be transforming for each subject in moving between these 2 modes of attention to reading, with close reading demonstrating, right, as we can see here, an almost global activation across brain regions. Ones that are reaching far, far beyond the ones conventionally associated with attention and executive function. So this is an example of an early data reconstruction with FSL. And what we're seeing is cross-sections of just one subject's brain for, in the case of close reading, right? And so, how many of you are less familiar with fMRI? Is everyone here to be pretty solid? Yeah. Okay, I'm going to do it. So basically, just to say, you know, the red areas here, sort of marking activation, right, are always working in comparison. They're not just marking activation, so to speak, in the brain, but areas where there's more blood flow in comparison to something else, right? And so here, red marks a significant increase in blood flow for close reading, right? As compared to pleasure reading, on average, over the total of the scan. And this subject, as we can see, is demonstrating surprisingly widespread changes for literary analysis across multiple regions, across multiple cross-sections, and unexpectedly broad areas of the brain. And this emerges with particular strength if we actually add another comparison to another subject. Obviously, we would expect another difference between reading something and not reading at all to be the far more drastic of the two comparisons. But in this subject, like almost all of the ones in our study so far, it's the top image, right, where we see the far wider or more global activations and differences, which is in fact shocking compared with what we see at the bottom, right, which is reading versus not reading at all. Reading versus not reading. So when they're in a moment of pause in between the text, they're not looking at any text at all, right, and that's acquired throughout the slices, throughout the scan. Yep. So again, to ask, right, so why care, right? And what this suggests, potentially, right, is it's the degrees of attention that we're paying in reading, right? Because we're comparing reading states, and they're always reading Jane Austen, that are activating these widespread parts of the brain and modulating these neural signatures, not just the stimulus, right, which has been the conventional sort of model of assumption. So again, what does this mean? Again, it's early, but if such results hold, which is truly what we're seeing so far, there are a number of potential implications. One, is the kind of controversy raging right now over the value of doing humanities, majoring in the humanities, studying it, and what this kind of preliminary research suggests, at least at its most basic level, is that this type of work has enormous cognitive complexity, right, at its very base. Moreover, it may not only be the books that we read, but the act of thinking or attending rigorously to them, the mode of focus that we bring to them that's of value, sort of integrating sort of diverse kinds of processes across the brain. And there's more. So still more interestingly, I think, we're not just seeing close reading activating more blood flow across the board than pleasure reading. Specific brain regions, again, we have to wait for cross-group analysis, but I wanted to give you this sort of, again, an individual sample from our study as a sort of case to sort of show why this is interesting. So red and yellow in this example mark increased activation for close reading, but blue marks increased activation for pleasure reading. And this is actually far wider in a lot of the subjects that we go through. So right here we see, for example, literary analysis activating unexpected regions we would not necessarily imagine seeing things that we use to place ourselves spatially in the world, such as the somatosensory cortex, right, and ways of sort of the motor cortex, an area largely dedicated to physical activity or imagining physical activity. Right, but we also, as I said, have the heightened activation for pleasure reading. So the question is, what do we do if so? And if so, close reading may not merely be a state of sort of heightened attention to literature, with pleasure reading a merely sort of lesser or more dormant mode of attention and focus, but instead to suggest that each style of reading has its own sort of cognitive demands and neural signatures and creates distinctive patterns in the brain. And so basically, since it's so early, I will sort of talk about some future directions. So the question is, what's next? And first of all, cross-group analysis. But in general, there are a lot of new studies and technological advances in neuroscience for the past few years that have been particularly important in moving us towards a richer understanding of the neural dynamics of reading. And I would point to Stanislaus Dehaene as a really good example of this for anyone who wants to dig more deeply. But in particular, rather than working to identify and isolate single regions of interest, people doing this work become increasingly attuned to neural complexity and the multiplicity of advanced cognitive processing, and thus able to handle these kinds of questions about literary and artistic engagement. So to take this example, one fMRI study in Japan, for instance, found that they could actually predict just from the brain images whether or not an art student had been looking at an artwork by Picasso or Dali. Things have grown still more interestingly, and this actually came up in our last talk a little bit, with the rise of a method known as functional connectivity. Rather than seeking to localize cognitive experience to a predefined brain region, such studies are interested, in fact, in the synchronous patterns in many ways, and sort of the patterns of connection that unfold or evolve as we engage with the stimulus, or for us, right, a work of art. And these experiments map then, right, not single regions, but activations and rhythms in the brain that emerge in parallel, right, across different regions of the brain. A lot of this grows out of resting state scan work. But one fMRI study, to give you a case, showed that rhythmic brain activations in storytellers and story listeners, for instance, actually become aligned over time as the storyteller and listener are engaging with the story. Synchronizing the two. And just to give you, I wanted to give you a final example of this, the power of this technique for reconstructions. I wanted to give this example of work from the Gallant lab at Berkeley. So as we can see, what we get is using complex algorithms for reconstructing fMRI data to create dynamic brain images of movie clips, right? Or as one of the speakers might put it, cognitive responses to them or representations of them. What we get as a result are unbelievably complex visual images of what people are viewing derived only from that subject's brain activation, right? As you can see, these remapped sort of brain images. I'll just do it again while I'm talking. Right, is that they match the color, shape, motion of the original images to an uncanny degree, right? In fact, mirroring the film's images as they move. And it's sort of this image of multiplicity and dynamism that in fact lies behind and motivates our next steps for the Austen study, which means there are a number of things, right, that we're looking at. So, as I said, cross-group analysis. But next, after that, some of the most interesting questions actually arise from integrating the eye tracking, the marks of what people are noticing visually with the final essays, what they attended to and remembered, and again, back to the brain images. And this can work because people, surprisingly, are actually quoting from the text in the essays that they write, which gives us something to tag and return to as part of the time course data. So the final and most daring goal, and this is far off, is in fact something like functional connectivity, using the eye tracking, in fact, to analyze attention over time, correlating the time-locked data for each reader, right, and doing something known as a time warp to produce a real-time view of reading Jane Austen over time. So I won't get into the details of that because we're fairly limited on time. But one of the things that's interesting about that, I think, for this group is that the reason it can work for moving images is because they have a stable T, or a stable time course, right? But in the case of reading, like with so much of how we process the world, we have vast individual differences. This means actually being able to time lock the eye tracking points of fixation, correlate them across a number of individual subjects, and then move back so you can actually get that kind of visual image of reading over time. So I'll end by talking about sort of concluding, finally, and perhaps most excitingly, this sort of fMRI of attention in Austen, recently known in the media as Your Brain on Jane, has inspired a series of new projects in our new lab, Digital Humanities and Literary Cognition. And this includes, and I'm happy to talk about more in the Q&A, but for now just to say, this includes an experiment, that's actually the second one, on trauma narratives and structures of empathy, another on poetry, music, and cognitive rhythm, that involves integrating fMRI and EEG, and a final one on actually looking at distractions in the reading, looking at differences when we read an iPad or Kindle as opposed to a traditional book. And this is supported by MSU, Stanford, and Lund in Sweden, supported by the Longberg Foundation. So just to talk more broadly, in all of these cases, I guess, to say my goal is to show that bringing together literature and neuroscience can in fact energize both fields, and that you can find points of translation between them and translators. But that this, in fact, adds a crucial piece to the larger puzzle and helps us understand the complex neural networks involved in reading aesthetics, engagement, and attention. And then more broadly, as someone crossing between humanities and the sciences, I'd like to end and suggesting that there's a sort of unique value in interdisciplinary experiment design. Because to talk about experiments like the ones I just described, with COPI in English and neuroscience, isn't just about the fMRI or its results. It's to talk about what it means to actually translate conversations like the ones we're having today into action. And in terms of this action, I guess I wanted to say what I'm seeing myself thus far, at least in the field of literary neuroscience. I think there's like 3 of us, are a number of interesting trends that really make this work possible. And I think most relevantly, as we think about this initiative at MSU, what I'm seeing is not only, right, these interdisciplinary experiment designs, but also interdisciplinary hubs, which are what, in fact, allow you to do this work, because if you need one thing, you need space. So we have a few models. There's a Center for Neurobiological Imaging at Stanford. There's a group called Neurohumanities at Duke. And then there's, at a microcosmic level, the DHLC and MSU. In addition, right, these being made up of cross-institutional groups of major players, like, right, producing experiments like those bringing together MSU, Stanford, and Sweden. And finally, and I think this is the result and the value of this kind of work, is that you see, by way of this, experiments that in fact integrate multiple tools and technologies in new ways. We were one of the first groups at Stanford, for example, to use the fMRI and fMRI-compatible eye tracking, in part because we were handling such a complex data set. So what I'd like to say in this final group is that these sort of experiments that integrate the multiple tools and technologies, be they fMRI and fMRI-compatible eye tracking, fMRI and EEG, or robotics and TMS, transcranial magnetic stimulation, that all of these are part of the key arguments and patterns and trends to consider as we consider the sort of infrastructures for not only for translating between neuroscience and humanities, but also for translating neuroscientific results into computational algorithms and AI, and envision the mind-brain work coming together across colleges and MSU. Thank you.
Take a few questions. Mike.
Yes.
How much kind of difference or bias do you find in terms of the level of sophistication of your trained experts? Because it seems like that even amongst them, there could be a wide variety of experience, interest, field.
No, and there's a lot of individual difference work, and that's actually part of why we're so excited about the eye tracking as well as the essays, which means we can actually correlate those markers of individual difference in training for the subjects as we continue to add them. It's not statistically significant at this point to do it that way because we have such a relatively small sample size, but that's kind of where it's heading in the future. And we did run in the pilot, sort of comically, that I used to use as a sort of joke slide, 2 neuroscientists who hadn't had any literary training. The activation was incredibly small. But the point, my point that I always would make, what? No, of course. Well, yes, or they're not alternating as much for the shooting and the pleasure reading. They said they had trouble with Austen. But basically, what we do is we actually get an incredibly sophisticated set of answers from them about how long ago has it been that they read Jane Austen, and what is their training, how long have they been working on this. And I think one of the things we'll be looking at in the future, right, is to be able to compare, right, those sort of levels of training, to be able to think about neurodevelopment in a new sort of way, and specifically about disciplinary training. A lot of people have been asking about law as well, mathematics, right? How does this compare to modes of reading, right? Yeah.
In terms of the image reconstruction from fMRI, could you say a little bit about the detail in terms of whether the system needs to be trained and how
it does. It does, and this is where I am indeed an English professor, so I won't be able to give this the most sophisticated answer I'd like to, but yes. So basically, what they need to do, from what I understand from my colleagues at Berkeley, is they do need to test each individual subject and then train the algorithm on that individual subject. So they need to check color. Right? We can imagine this as the tagging, that it would take to create that reconstruction. So something about color, something about motion, certain objects, and then you begin to create a really complex matrix for then reconstructing those images back from those individual subjects. The same is happening with the Picasso and Dali study. It needs to be trained on the individual subject, but I do think that's where some of the most interesting work is going on. This is where I also would like to have the outreach, because there is a limit to how much I can learn as an 18th century English professor. But I think these questions about what would you tag, for example, in a complex work of literature, where you don't have that kind of visual stimulus. You're going to tag metaphor. Are you going to tag, right? When we get towards these really complex aspects of artificial intelligence and AI, and I think that dealing with the complexities of language, not just sort of character recognition, but how we read, then maybe begin to think about these sorts of tagging as the ongoing process. Yeah.
How much do you think that this overactivation during close reading actually has to do with reading at all? Will they get subjects look at stimuli that look vaguely like letter forms, but are not, don't represent words, and ask them later to recall what they remember?
Yeah, so we did not do a sort of Jabberwocky control, right? In part because there were just too many variables floating around, because it would have just scrambled it. But basically, the one thing I was most concerned about was that we would simply see, activation in regions associated with anxiety, right, with pressure and testing, and it's clearly not that. It's not just basis. It's beyond that by a large margin. So I think that'll be one of the things that will emerge as we do. Then we're going to do another round at MSU, and so if you have any ideas for how to incorporate that, I'm really thinking about how to adjust the experiment as needed to handle some of these questions. It's a difficult one because you need to have enough time to get the anatomical scans and then to do a relatively long reading task. So having them do anything statistically significant before that gets challenging in the scanner. So there's a little bit of that, as always, payoff in terms of your complexity and the amount of time you can keep them inside a loud MRI. Yeah.
While Karim is setting up,
We have
time
one more, maybe? Yes?
I just wondered if you take into consideration learning style, because some people are readers, some people are visual, some people, can't read if they have cognitive disabilities. So do you take that into consideration, or do you test for that?
We don't test for that in particular. We do ask about particularly developmental questions, so we did not say anyone who had dyslexia, attention deficit, things like this, things that were pertinent to the study. It's amazing how few English PhDs, though, have no type of anyway, but basically, what we are doing instead of that is looking at the essays. I think this is where this will become quite rich, to reverse engineer that kind of question. In other words, to look through and say, are they focusing on visual imagery? Are they focusing on things that are tactile? Are they processing this more conceptually? Who's talking about the plot? Who's talking about, right? And then being able to use those essays, again, to sort of like, in a way, the sort of managing the complex big data of brain axes in a way on a small scale, right? With these infinitely complex aspects of qualitative data. So the question is simply, how do you map them in reverse? Yeah, that's an excellent question.
All right, well, let's thank Natalie again.